Hi everyone, this is Dr. K. Prabhu. In this video, we got to discuss about polymerase chain reaction. We got to discuss about the introduction of PCR, components of PCR, steps involved in PCR, key factors in PCR, and its application. PCR is an efficient and cost-effective molecular tool to copy or amplify small segments of DNA or RNA. It's a molecular technique to create several copies of certain DNA segment. This technique was developed in 1983 by Carey Mullis, an American biochemist. PCR combines the principle of complementary nucleic acid hybridization with those of nucleic acid replication that are applied repeatedly through numerous cycles. It results the exponential production of the specific target DNA or RNA sequences by factor of 10 to the power of 7 within a relatively short period. This is in vitro amplification. Amplify a single copy of nucleic acid and target by using two synthetic oligonucleotide primers that bind to the target genomic sequence, which are extended by a TAQ polymerase. TAQ polymerase is a thermostable DNA polymerase. So it is an automated process of repeated cycles, usually 25 to 40, of denaturation of the template DNA, annealing of primers to their complementary sequences, and primer extension. These three steps happen in different temperatures is employed to amplification of the target sequence. PCR was originally developed in 1983 by American biochemist and is a Nobel laureate Carey Mullis. What is the principle behind? Before we go to the components, what is the principle behind in PCR? The PCR technique is based on the enzymatic replication of DNA. In PCR, short segment of DNA is amplified using primer-mediated enzymes. DNA polymerase synthesize new strands of DNA complementary to the template DNA. The DNA polymerase can add nucleotide to the pre-existing 3' hydroxyl group only. Therefore, a primer is required. Thus, more nucleotides are added to the 3' end of the DNA polymerase. Now we back to the components of polymerase chain reaction. DNA template, deoxyribonucleoside triphosphate, PCR buffer, primers, both forward, backward, TAQ polymerase. First one, primer, a short segment of nucleotide which is complementary to the section of DNA or RNA which is to be amplified in the PCR. Two short DNA sequences designed to bind to the start and end of the target sequences is used in PCR. TAQ polymerase, Thermus aquaticus polymerase is a thermostable DNA polymerase originally isolated from the thermophilic bacterium Thermus aquaticus, which resists inactivation during the denaturation temperature and allows the primer extension to high temperature. So next topic is the steps involved in PCR. To perform PCR, extracted sample which contains the target DNA template is added to a tube containing primers, free nucleotides, and TAQ polymerase. The PCR mixture is placed in a PCR machine. PCR machine increases and decreases the temperature of PCR mixture in automatic programmed steps which generate copies of the target sequences exponentially. Polymerase chain reaction has three major steps as we already discussed: denaturation, annealing, extension. The first one is denaturation. Is a strand separation is happening in the denaturation. The separation of the two hydrogen bonded complementary chains of a DNA into a pair of single-stranded polynucleotide molecules by a process of heating at 94 degrees Celsius to 96 degrees Celsius. The second step is annealing. It's a primer binding step. The temperature is lower to 45 to 60 degrees Celsius, so the primers can attach themselves to the single-stranded DNA strands. The third step, the new DNA was synthesized step. It is shortly called as extension step. It stored the annealed primer and work its way along the DNA strand, and the step is happen at 72 degrees Celsius. Once the first round is completed, the process is repeated by cycling back to the first reaction temperature, and next round to denaturation, annealing, and extension is started. So, as a automatic process in thermocycler, so this three steps temperature cycle is repeatedly approximately 30 times, which results exponential amplification of a target gene sequence. So this is the illustration about the three steps. The first step, 1 minute. The second step, 1 minute. Third steps happen in 2 minutes to amplify the target DNA. So as we already told, it is an automatic process. So at the end of the 30 PCR cycles, we get 1073741824 number of copies of target DNA. The next topic is the key factors for optimal PCR. See, primer is very important, DNA polymerase is very important. Each and everything involved in PCR is important for getting the proper amplification. The primer 70 to 30 nucleotide contained sequence without secondary structure and complementary among themselves is an ideal one. The complementary primers can uprise to form primer dimer and get amplified in PCR, thereby preventing the multiplication of target DNA. DNA polymerase, the second important key factors. TAQ DNA polymerase is preferred as it can be withstand high temperature. In the hot start protocol, DNA polymerase is added after the heat denaturation step of the first cycle to avoid the extension of mismatched primers occurring at the low temperature. TAQ polymerase lacks proofreading exonuclease activity contributing the error in the PCR products. TMA Thermotoga marittima DNA polymerase and PFU. PFU is nothing but Pyrococcus furious. Its DNA polymerase have been identified with a thermostable and proofreading activity. Third factors target DNA. The shorter the sequence of target DNA, the better efficiency of PCR. In recent years, amplification of DNA fragments up to 10 kb has been reported. The GC-rich regions of DNA strand hide into the PCR. Fourth one is a promoter. Addition of proteins such as BSA enhances the PCR performance by protecting DNA polymerase. The fifth key factor is inhibitors. Humic acids frequently found in archaeological samples of target DNA inhibit PCR. So next one is applications of PCR. So following are the applications of PCR in medicine: testing of genetic diseases, mutation, monitoring the gene in gene therapy, detecting diseases causing genes in the parents, in forensic science used as a tool in genetic fingerprinting, identify the criminal from millions of people, paternity tests in research and genetics, compare the genome of two organisms in genomic studies, in the phylogenetic analysis of DNA from any source such as fossils, analysis of gene expression, and gene mapping. Thank you for listening.